Hello students, today let us study about the topic biofertilizers under the main subject agrochemical and pest control. Biofertilizers are not fertilizers. Biofertilizer is a substance which contains living microorganisms which, when applied to seed, plant surfaces or soil, colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plant. Biofertilizers add nutrients through the natural process of nitrogen fixation, solubilizing phosphorus and stimulating plant growth through the synthesis of growth promoting substances. Biofertilizers can be expected to reduce the use of chemical fertilizer and pesticides. The microorganisms in biofertilizers restore the soil's natural nutrient cycle and build the soil organic matter. Through the use of biofertilizers, healthy plants can be grown while enhancing the sustainability and the health of the soil. Hence, in this episode, the following aspects are going to be seen. They are types of biofertilizers, manufacture of biofertilizer by the indoor method, applications of biofertilizers to crops, advantages of using biofertilizers, and finally, disadvantage and limitations of biofertilizers. Let us study these aspects one by one. The first aspect, types of biofertilizers. Biofertilizers are classified as nitrogen biofertilizers, compost biofertilizers and phosphorus biofertilizers. Let us study about them briefly one by one. Nitrogen biofertilizers. This type of biofertilizer helps the soil to correct its nitrogen level. Nitrogen is an essential component for plant growth, but plants need it in a limited amount. Different soils have different requirement for nitrogen. That is why it depends on the cultivated crops that which type of nitrogen biofertilizer should be used. Azotobacter is a biofertilizer which provides the required amount of nitrogen to the plant from the soil. Reduction of dinitrogen through biological means is termed as biological nitrogen fixation. Though atmosphere contains 79% of nitrogen, eukaryotes cannot utilize the nitrogen gas directly unless it is reduced to ammonia. Dinitrogen having triple bond and cannot be broken by higher plants. Broadly, the nitrogen biofertilizers is divided into two groups. They are symbiotic and non-symbiotic. Symbiotic nitrogen fixation occurs in plants that harbor nitrogen fixing bacteria within their tissues. The best studied example is the association between legumes and the bacteria in the genus Rhizobium. Each of these is able to survive independently, which means the soil nitrates must then be available to the legume. But life together is clearly beneficial to both. Only together can nitrogen fixation take place. A symbiotic relationship in which both partners benefit is called mutualism. Symbiotic nitrogen fixation may be of three different types. They are legume rhizobium, cyanobacteria with plant or fungi, and frankia with trees. Rhizobium. Rhizobium is symbiotic bacterium. It occurs in the roots of different legumes and pulses, produce nodules and fix atmospheric dinitrogen inside the nodules. This rhizobium present inside the nodule can be isolated and multiplied in laboratory. After appropriate multiplication, the rhizobium is mixed with carrier material in aseptic condition and used as biofertilizers for different pulse crops. Next is cyanobacterial associations. The photosynthetic cyanobacteria often live as free living organisms in pioneer habitats such as desert soils or as symbionts with lichens in other pioneer habitats. They also form symbiotic associations with other organisms such as the water fern Azolla and cycads. The association with Azolla, where cyanobacteria are harbored in the leaves, has sometimes been shown to be important for nitrogen inputs in rice paddies, especially if the fern is allowed to grow and then plowed into the soil to release nitrogen before the rice crop is sown. A symbiotic association of cyanobacteria with cycads shows a pot-grown plant. The second image shows a close-up of the soil surface in this pot. Short club-shaped branching roots have grown into the aerial environment. These aerial roots contain a nitrogen-fixing cyanobacterial symbiont. Next is Frankia with trees. Frankia is a genus of actinomycetes that has the capacity to fix nitrogen gas in association with several tree species. Examples of such trees include the genera Casuarina and Allocasuarina in the family Casuarinaceae and Alnus in the family Betulaceae. The associations formed by species of Frankia are generally host plant specific, but the same bacteria can nodulate closely related plant species. Frankia associations with plants range from those that are highly effective to those that are relatively ineffective at fixing nitrogen. Nitrogen deficiency in Casuarina can be remedied by inoculation with strains of Frankia that are effective at fixing nitrogen. Biological nitrogen fixation performed by a group of autotrophic bacteria living free in the soil either aerobically or anaerobically and not dependent on plants. Non-symbiotic nitrogen fixation can be divided into another three groups based on the oxygen requirements. They are aerobic free living nitrogen fixation, example Azotobacter and Bergeringia. Microaerophilic, for example Azospirillum, and anaerobic free living, for example Clostridium and Desulfovibrio. In all the nitrogen biofertilizers system, it is the nitrogenase enzyme that play a crucial role in reducing dinitrogen to ammonia. Compost biofertilizers are animal waste which are degraded by the bacteria and used as the best naturally occurring biofertilizers. They not only protect the plants from diseases, but also help them to grow in a healthy environment. Composting is one of the oldest techniques used for the stabilization of natural waste and soil biologic fertilization. The main objective of this practice is to obtain a stable, chemical and biological rich product with micro and macronutrients. The composting process works as follows. Initially, strains of microorganisms break down living waste, generating temperature differentials, while the pH of the medium decreases as a result of the production of natural acids. Once the temperature gets close to 40 degrees centigrade, thermophilic bacteria initiate degradation processes, making the temperature to reach 65 degrees centigrade. Under these conditions, the metabolism of certain fungi is deactivated. During this stage, biological transformation reactions are developed by actinomycetes and fungi spore forming bacteria. These quickly consume easily degradable compounds such as sugars, proteins, starch and fat. In addition, the pH tends to be alkaline due to the release of ammonium ion. Once degraded the organic material, the reaction rate decreases as well as temperature. This stage is known as cooling. Both thermophilic and mesophilic fungi are capable of degrading cellulose during this phase. Finally, ripening process begins, which requires several months, at least 4, to be completed. This stage can lead to the complete degradation of compounds and to obtain stable material. The composting process can lead to obtain stable humus and humic and fulvicin acids characterized by high nutritional value and potential for fertilization of soils with nutrient deficiencies. Vermicomposting. Vermicomposting is a biological fertilization technique consisting of the use of earthworms metabolism to produce humus with high nutrients content. To apply it, organic waste is required. Example, manure, fruit peel and crop residues. The organic material passes through the digestive tract of worms, where it is transformed into a material rich in microorganisms, macro and micronutrients. Based on that process, a chemical and biological stable fertilizer is obtained. This technique can be developed and applied successfully at both small and large scale in various environments or under controlled laboratory conditions. Phosphorus biofertilizers. Phosphorus is also a limiting factor and plants need it in particular amount. Phosphorus biofertilizers help the soil to correct the phosphorus level. As nitrogen biofertilizers depend on the cultivating crops, phosphorus biofertilizers do not depend on the cultivating crops. Rhizobium is the phosphorus biofertilizer. Moving on to the second aspect, that is manufacture of biofertilizer by the indoor method. The aim of the indoor method of manufacturing compost is by a means of simple process to unite the advantages of 3 very different things. They are the result of scientific research on the transformation of plant residues, the agricultural experience of the past, and the ideal line of advance in the soil management of the future in such a manner that all the byproducts of agriculture can be systematically converted into humans. An essential feature of this synthesis is the avoidance of anything in the nature of fragmentation of the factors. All available vegetable matter, including the soiled bedding from the cattle shed, all unconsumed crop residues, fallen leaves and other forest waste, farmyard manure, green manures and weeds pass systematically through the compost factory. The only other materials employed are air and water. This manufacture is continuous right throughout the year, including the rainy season when a slight modification has to be made to ensure sufficient aeration. The product is a finely divided leaf mold of high nitrifying power ready for immediate use. The fine state of division enables the compost to be rapidly incorporated and to exert its maximum influence on a very large area of the internal surface of the soil. The indoor process thus utilizes all the byproducts of agriculture and produces essential manure. Besides doing this, any successful system of manufacturing compost must also fulfill the following conditions. The first condition, the labor required must be reduced to a minimum. The process must fit in with the care of the work cattle and with the ordinary working of the farm. Next, a suitable and also a regular carbon nitrogen ratio must be produced by well mixing the vegetable residues before going into the compost pits. Unless this is arranged for, decay is always retarded. The mixing of these residues combined with the proper breaking up of all refractory materials is essential for rapid and vigorous fermentation and for uniformity throughout the process. Next, the process must be rapid. To achieve this, It must be aerobic throughout and must include arrangements for an adequate supply of water and for inoculation at the right moment with the proper fungi and bacteria. The general reaction of the mass must be maintained within the optimum range by means of earth and wood ashes. The maintenance of the proper relationship between air and water so that no delay takes place in the manufacture proved to be the greatest practical difficulty when evolving the process. Next, there should be no losses of nitrogen at any stage. If possible, matter should be so arranged that fixation takes place in the compost factory itself and afterwards in the field. To conserve the nitrogen, the manufacture must stop as soon as the compost reaches the nitrification stage, when it must either be used for banked. It can be best used as a top dressing for irrigated crops. It can be preserved as money is kept in a bank by applying it to the fields when dilution with a large volume of soil arrests further changes till the next rains. Next, there must be no serious competition between the last stages of the decay of the compost and the work of the soil in growing a crop. This is accomplished by carrying the manufacture of humus up to the point when nitrification is about to begin. In this way, the principle of dividing the growing of a crop into two separate processes, that is, the first process, the preparation of the food materials outside the field, and the second, the actual growing of the crop, can be introduced into general agricultural practice. Next, the compost should not only add to the store of organic matter and provide combined nitrogen for the soil solution, but it should also stimulate the microorganisms. Next, the manufacture must be a cleanly and a sanitary process from the point of view both the man and also of his crops. There must be no smell at any stage. Flies must not breed in the compost pits or in the earth under the work cattle. The seeds of weeds, the spores of harmful fungi, the eggs of noxious insects must first be destroyed and then utilized as raw material for more compost. All this is achieved by the combination in the compost pits during long periods of high temperature and high humidity with adequate aeration. Moving on to the third aspect, that is applications of biofertilizers to the crops. The biofertilizers can be applied to the crops in different forms. They are seedling root dip. This method is applied to the rice crop. A bed of water is spread on the land where the crop has to grow. The seedlings of rice are planted in the water and are kept there for 8 to 10 hours. The next is seed treatment. In this method, the nitrogen and phosphorus fertilizers are mixed together in the water. Then the seeds are dipped in this mixture. After the application of this paste to the seeds are dried. After they dry out, they have to be sown as soon as possible before they get damaged by harmful microorganisms. Next is soil treatment. All the biofertilizers along with the compost fertilizers are mixed together. They are kept for one night. Then the next day, this mixture is spread on the soil where seeds have to be sown. Moving on to the fourth aspect, that is the advantages of using biofertilizers. Biofertilizers are becoming a rage considering the irreparable damage that the chemical fertilizers are causing to the soil. Some of the advantages associated with biofertilizers include: the first and the most important advantage of using biofertilizer is that they are environment friendly, unlike chemical fertilizers that damage the environment. They are comparatively low on cost inputs and are light on the pockets of the farmers. Their use leads to soil enrichment and the quality of the soil improves with time. Though they do not show immediate results, but the results shown over time are extremely spectacular. Microorganisms convert complex organic material into simple compounds so that the plant can easily take up the nutrients. These fertilizers harness atmospheric nitrogen and make it directly available to the plants. They increase the phosphorus content of the soil by solubilizing and releasing unavailable phosphorus. Biofertilizers improve root proliferation due to the release of growth promoting hormones. They help in increasing the crop yield by 10 to 25%. Disadvantage and limitations of biofertilizers. Applying biofertilizers is unlikely to harm plant life or the environment in any way, but there is little to guarantee that they will help either. Compost products have highly variable concentrations of nutrients. In addition, implementation costs are higher than those of certain chemical fertilizers. Extensive and long-term application may result in accumulation of salts, nutrients and heavy metals that could cause adverse effects on plant growth, development of organisms of the soil, water quality and human health. Large volumes are required for land application due to low contents of nutrients in comparison with chemical fertilizers. Main macronutrients may not be available in sufficient quantities for growth and development of plants. Nutritional deficiencies could exist caused by the low transfer of micro and macronutrients. Finally, to conclude with today's topic, the relevance of biofertilizers is increasing rapidly since chemical damage the environment. In contrast, biofertilizers lead to soil enrichment and are compatible with long-term sustainability. Further, they are eco-friendly and pose no danger to the environment. Thank you.